Yes, I'd like to thank the organizers for giving me this opportunity to share with you some of our work. As Tanisha mentioned, we are primarily interested in the switch-SNF chromatin remodeling complex, and I realize many of you may not be familiar with this complex, so I thought I'd go through some of the background on its role in embryonic development and take you through sort of newly appreciated studies confirming its role in cancer, and then dive into some of our mechanistic analysis on the mode of action of these complexes. So in my lab, we're primarily interested in the epigenetic regulation of gene expression. So for example, it's known in stem cells that many lineage-specific genes are kept in this repressed conformation in compacted chromatin. But upon the receipt of differentiation stimuli, multiple mechanisms contribute to the activation of this gene. And so including DNA demethylation, the acquisition of histone modifications, as well as this sort of large-scale changes in chromatin structure. And it's really this nucleosome decompaction, this event of taking inaccessible chromatin to accessible forms that we're most interested in, commonly referred to as chromatin remodeling. And so in particular, we're interested in this switch-SNF chromatin remodeling complex. This is a large 2 megadalton multi-subunit complex consisting of a core ATPase, either BERG or BERM, and up to 14 other associated subunits. And importantly, many of the subunits in this complex are members of gene families, and these homologs can kind of alternately substitute for each other, giving rise to a diverse array of potential switch-SNF complex variants. And it's known that this combinatorial assembly is critical in the context of cellular differentiation. So for example, in neural progenitors, a certain assembly of subunits is expressed, and during neuronal differentiation, several of these subunits are switched out for homologous forms to give rise to the sort of neuronal switch-SNF complex. And if you perturb the switching by either prolonging the expression of these subunits or deleting the expression of the neuron-specific subunits, you completely dysregulate neuron differentiation. So it's not only required for cellular differentiation, but it's also instructive. So for example, if you express components of the stem cell switch-SNF complex in fibroblasts, you can enhance the ability of these cells to become reprogrammed. Likewise, if you express the neuron components in fibroblasts, you can actually induce transdifferentiation into induced neurons. So it's both sort of has this necessary and sufficient quality for cell lineages. And this is not just in the context of neuronal differentiation, but it's now known that switch-SNF subunits have a variety of cell type and developmental stage-specific activities in multiple different lineages, including the very earliest stages of embryonic development. So in very early studies, it was shown that this complex may have tumor suppressor activity and that many human cancer cell lines lack expression of these subunits. And if you reconstitute the complex in these cell lines, you basically curtail cell growth, indicating that this complex may have some tumor suppressor action. So fast forward 20 years and many sequencing studies later, we now have this very sort of rich resource of through the CBio, where we can now appreciate that these genes are mutated in a large array of tumor types. So this is simply the mutation frequency of all switch-SNF subunit genes across each of these different cancer types. And we now estimate that over 20% of human cancers have a mutation in one of these genes. So very important and sort of newly appreciated function. Statistically speaking, this is not just passenger mutations, because if you look at the sort of fraction of patients with a mutation versus the significance of this calculation, you find that switch-SNF subunits are mutated as significantly as some tumor suppressors such as P53, APC, PTEN. So this is our current understanding of the role of the complex in cancer. And again, I'd like you to appreciate sort of the context-specific roles of each of the subunits. And in particular, we really have no mechanistic understanding of the role of these subunits in BAF complex function or switch-SNF complex function. And so we were primarily interested in studying this one subunit ARID1A, which is the most frequently mutated subunit. It's mutated in 10 to 60% of adult tumor types, including ovarian, endometrial, gastric, bladder, hepatocellular, pancreatic, colorectal, breast, and lung. So predicted to affect a large number of individuals. But in contrast, its homolog ARID1B is very rarely mutated in cancer. So we wanted to understand what was sort of the defining principle of all of these different types of mutations. And one thing that has come to light is sort of this synthetic lethality between homologs. So in the context of a particular switch SNF mutation, it is often the residual homolog that forms a stable complex that is required for cancer cell proliferation. So for example, in the context of an ARID1A mutation, ARID1B is required. In the context of a SMARCA4 mutation, its homolog SMARCA2 is required. In the context of a SMARCB1 mutation, there's a fragment that's expressed that is essential. And even in the context of some switch SNF wild-type tumors, the complex has been shown to be critical for cell growth. So this has given rise to a lot of excitement around the development of switch SNF complex inhibitors, both to target switch SNF mutant cancers as well as, in some contexts, switch SNF wild-type cancers. So we really wanted to understand mechanistically what is the tumor suppressor mechanism, particularly focusing on ARID1A, which again is the most frequently mutated subunit, and what is the sort of synthetic lethal phenotype that underlies these homologs. So we envisioned a couple of different scenarios for this. One possibility is that ARID1A and ARID1B may have play redundant roles for an essential function, with ARID1A having non-redundant roles in tumor suppression. So then in the context of an ARID1A mutation, ARID1B would then be required to fulfill this essential function. It's also possible that ARID1A and ARID1B may have non-redundant roles, and this has been shown, for example, in different regulatory interactions formed by ARID1A and ARID1B containing complexes that allow them to be tumor suppressive or growth promoting. So then in the context of an ARID1A mutation, you may sort of disrupt this balance, allowing ARID1B complexes to kind of promote growth in an unchecked fashion. It's also possible that ARID1B complexes may undergo stoichiometric changes or different regulatory interaction changes that are specific to the ARID1A mutant setting. So to understand this, we really went back to our knowledge of the complex and its most basic function in chromatin remodeling, and this idea that it really serves the purpose of pushing these nucleosomes out of the way to allow accessibility in chromatin structure. And we employed this technique that, relatively recent technique called attack-seq, which basically relies on a transposase to deliver DNA adapters into accessible chromatin. And this can be easily amplified and sequenced to give these types of plots. And this has been optimized for 50,000 cells and even now down to the single cell, and gives information equivalent to sort of DNA hypersensitivity. So to understand mechanistically what's going on, we relied on isogenic cell lines that are either wild type or mutant for ARID1A. And so when we knock down ARID1B in the wild type setting, we see no effect on cell growth. However, when we knock down ARID1B in the ARID1A mutant setting, we now see a profound loss of cell proliferation, sort of recapitulating the synthetic lethal phenotype of these homologs. So we performed attack-seq in these two different settings. So this is just in the wild-type context here, and right away we had a very gratifying result that we had basically uncovered the mechanistic selective advantage for ARID1A mutations in the cancer setting. So in the context of an ARID1A knockout, we saw that thousands of sites changed in their accessibility genome-wide. Nearly 2,000 sites gained accessibility, nearly 7,000 sites lost their accessibility. However, knockdown of ARID1B had really no effect. And so I think this sort of underlies mechanistically why ARID1A is selected to be mutated in cancer. However, now when we knock down ARID1B in the ARID1A knockout setting, we now recover a role for ARID1B. So an additional 3,000 sites gain accessibility, an additional 5,000 sites lose accessibility. Again, mechanistically sort of recapitulating the relationship of these homologs in cell culture. So we wanted to understand whether ARID1B regulates ARID1A dependent sites or has a unique signature. So we basically clustered our accessible sites based on the degree of accessibility in the wild type setting, and then clustered all the genotypes up here. And we found several clusters that increased accessibility, as well as a large chunk that exhibited unchanged accessibility. But I'd just like you to focus on these two clusters in which we lost accessibility. And in this context, loss of ARID1A resulted in a slight reduction, which was then further augmented by the loss of ARID1B. indicating that ARID1B is largely compensatory in its function for maintaining chromatin accessibility. So we wanted to understand what makes these sites particularly vulnerable to loss of these subunits. So we mapped the binding of switch-NIF complexes against our accessible sites and found that switch-NIF binding is highly enriched in these sites that are particularly sensitive to ARID1A and ARID1B loss. So this is just to sort of summarize our findings here. We found that ARID1A has a dominant function in maintaining accessibility. The role of ARID1B is really only apparent in the absence of ARID1A, underlying this sort of synthetic lethal relationship. The dependency on ARID1A and ARID1B is defined by high switch-NIF binding, and we find that ARID1B is largely compensatory in its function. So given that the complex binds upwards of a third of all genes at both promoters and regulatory elements, we wanted to understand where in the genome these sites are being affected. So we overlaid our differential accessible sites with histone modifications that define either enhancers or promoter elements. And we found that our sites that lost accessibility were highly enriched for H3K27 acetylation and H3K4 monomethylation, but not enriched for H3K4 trimethylation, which is indicative of promoters. And so if we simply kind of dive in a little deeper here, we find that unchanged sites have a nice distribution of promoters and enhancers, whereas our decreased sites are really enriched at these enhancer elements of all classes, the poised, active, and super enhancers. And increased sites are primarily found at promoters. So we wanted to understand whether, in addition to regulating chromatin accessibility, these complexes also regulate active histone marks at enhancers. So we performed ChIP-seq for H3K4 monomethylation, H3K27 acetylation, and H3K4 trimethylation in each of the 4 genotypes. And we find that sites that lose accessibility also lose both K4 monomethylation and K27 acetylation in a stepwise fashion. H3K4 trimethylation is low at these sites, consistent with them being found at enhancers, and this modification generally does not change. So we can further go into our attack-seq data and define what kind of motifs are underlying these accessible regions. And we found that the members of the AP1 complex are particularly enriched. Those motifs are particularly enriched at sites that lose accessibility. And we can further parse our attack-seq data to isolate mononucleosome fragments that allow us to position nucleosomes around this motif. And we see that with the loss of ARID1A and ARID1B, we not only see a reduction in accessibility, but we see a reduction in nucleosome spacing, indicating that these nucleosomes really collapse in around this transcription factor motif and occlude it for transcription factor binding. And to really test this directly, we performed a ChIP for one of the AP1 complex members, FRA1, and we found that loss of ARID1A and ARID1B basically abolishes the ability of this transcription factor to bind its motifs. So this is, in summary, what we think is going on. We have estimated that ARID1A complexes are about 7-fold more abundant than ARID1B complexes. We find that they are primarily enriched at enhancer elements where they maintain chromatin accessibility, these active histone marks and transcription factors, as well as eRNA synthesis that I didn't show you. And then upon loss of these factors, you get sort of collapse of these regions, potentially impacting gene expression. So to understand what genes are particularly affected, we performed RNA-seq in each of our 4 genotypes. And as expected, we saw very little change with the loss of ARID1B in the wild-type context. However, loss of ARID1B in the ARID1A mutant context dramatically enhanced sort of the ARID1A knockout signature. And we found that the genes that were particularly enriched are genes in these receptor tyrosine kinase pathways. So not only the receptors themselves, but the ligands, the downstream signaling mediators, and transcription factors, leading to these sort of changes in cell cycle genes. And in particular, what we noticed was GO pathway terms for EGFR, the EGFR pathway and the EGFR inhibitor resistance pathway, because not only was EGFR itself and its ligand downregulated, but essentially all other receptors in this family were downregulated. So essentially indicating that by hitting this very downstream element, we really wiped out a whole host of different growth pathways that these cells might rely on. So it's often difficult to really connect enhancer regulation to transcription at the level of gene. So to connect all of these dots and to really emphasize the role of the ARID1A and ARID1B in regulating enhancers impacting gene expression, we overlapped all of our sites to find sites that had decreased accessibility, decreased K27 acetylation, binding of the complex, and binding of FRA1. And we found that over 350 downregulated genes had at least one of these sites in its proximity or regulating. So to drive this point home, we overlapped our data with an RNA Pol2 chia PET, which identifies interacting regions of DNA that are bound by Pol2. And we found that these sites that had these features of co-regulated down were more likely to interact with genes that decreased expression than genes that increased expression. And the vice versa was also true. So sites that had increased accessibility and increased K27 acetylation were more likely to interact with genes that were going up than genes that were going down. So this is just to give you an example of some of the browser tracks for what we've observed. So this is the MET gene encoding the MET receptor tyrosine kinase. So we found several areas in this gene where we see a high degree of accessibility, which is bound by FRA1, bound by the complex, has high levels of K27 acetylation, as well as nascent transcription, indicating that these sites are regulatory enhancer elements. And again, they exhibit this looping back to the promoter, suggesting that they can directly regulate transcription off the transcription start site. In the absence of ARID1A and ARID1B, we see a complete loss of accessibility at these enhancers, but not really at the promoter, and a complete abolishment of K27 acetylation. And this does indeed affect both the protein and gene and protein expression. So to see whether this is actually important, we tried forcing the expression of MET in our double knockout cells to see whether this could rescue the synthetic lethal phenotype. And we find that forced expression of MET has a very small but significant increase in cell growth, indicating that the rescue of just a single gene is probably insufficient to completely rescue this growth phenotype. So in summary, we've identified what we think is an essential function for the complex in maintaining chromatin accessibility. We feel that ARID1A and ARID1B participate in this essential function, and that in the absence of ARID1A, ARID1B kind of maintains this chromatin accessibility, which is particularly important for regulating enhancers. ARID1A may have additional non-redundant roles in tumor suppression, such as antagonism with polycomb group complexes, recruitment of HDACs, other things that have been implicated in this. So in summary, our data really argue for the development of switch-NF complex inhibitors to treat these cancers. We see global reductions with multiple growth pathways affected. Many of the aspects of our work we found to be conserved in a different cancer type of ARID1A immune cancer, including the sensitivity of enhancers, cooperativity with AP1 complexes, and the PI3 kinase being affected. And importantly, we think our data has implications for resistance in that no single gene was sufficient to rescue the synthetic lethal phenotype, and there may be little recourse for resistance in general upon development of these inhibitors. So with that, I'd just like to thank the people that contributed to this work. This work was just recently published in eLife. It's about to come online, and I'd like to thank my postdoc, Timothy Kelso, for really driving this work with help from Devin Porter and my funding sources. Thank you.
So besides the motif enrichment analysis, is there evidence for physical recruitment, even transient, like with apex fusions?
Yes, so there is a physical interaction between the complex and AP1 complex members.
And what about others? Runx family members, for instance, and ETS family members have been implicated in nucleosome repositioning through BRG-BRM as well.
I don't know offhand, but it's possible. I think yeah, we need to check that.
I'm actually surprised that you didn't pick up Runx in your motif analysis.
Really? Yeah, we have uncovered Runx in the context of a neural development pathway. Yeah.
So
yeah.
the cells you used, which I don't know what they are, don't express Runx?
Yes, so it's a colorectal cell line. Yeah, so we expect these to be these cooperative to be context dependent. Yeah.
guess we move on. So in terms of context dependent, are you picking up sort of a core program that might be conserved across cancers, or have you sort of looked at that?
Yeah, so we've looked at that by comparing multiple different cell lines. We do think that this is conserved, and we think particularly the interaction with AP1 might be something that we would pick up on, particularly in the cancer type. But we think that this also underlies the dependence of the complex in embryonic development. So multiple different settings where we feel this enhancer sensitivity is really the major essential role of the complex. So yeah.